Hi, this is Dr. Jabali, and in this video we're going to focus on topic 9.2, which is all about flow and translocation. Here are some of the things that we will target. We're going to talk about how organic compounds such as glucose, for example, is going from the sources to the sinks. We're going to talk about how water is so important in doing the hydrostatic pressure gradients, pushing the food along the plant. We're also going to talk about the act of transport, putting the organic molecules into the phloem. We're also going to talk about how concentrated solute has important effect in the phloem, as well as the water going in as osmosis. We're going to talk about the hydrostatic pressure causing the phloem to flow towards the sink. We're going to talk about the structure and the functions of the phloem, as well as identification of the xylem phloem in the microscope image. Besides that, we're also going to look at the aphid stylets and look at how them using radioactive-labelled carbon dioxide, you can see the rate of the phloem transport. So with that, let's learn a little bit more about the phloem. Phloem is basically one of those vascular bundles that are just putting nutrients up and down. So as you can see here, it's where it usually carries also photosynthesis. You can have the leaf with all the sugars being produced. Now, the movement here is always up and down, and it's alive during maturity, unlike the xylem. If you remember, the xylem, it's actually dead when it's mature. Now, here you also have the two pieces of the phloem, right, which is known as sieve tube and companion cells. Those are the two pieces. The sieve tube is more perforated. That means it has holes in the walls that are very thick and rigid, and there is no nucleus whatsoever. Okay, so in this case, we're talking about here the sieve tube. You can see these holes, and this whole area here that I'm highlighting is very thick and rigid, and you can see there's no nucleus. Now, companion cells, on the other hand, they have mitochondria, and it's important because there's a lot of active transport in their cell membrane. And they also have a lot of enfolding walls because they want to increase the surface area, and active pumps, as I said. So, companion cells is where you have, again, membranes that are made enfolded. You have the active transport, which it's a protein channel that basically needs ATP, and for that reason you have mitochondria. So both sieve tube and companion cells, they make the phloem. Now here is just to understand the connection between the phloem as well as, the companion cells with the sieve tubes. There's plasma nata, which is basically their cytoplasm kind of connected. Now, if you had to draw this, you literally would have been drawing right here the companion cells, and then you do a little circle right here because you have to have the nucleus, and then you draw a circle like this, and you do a line, and you connect with little circles here, and that will be the sieve tube. So those are the two pieces that makes the phloem. Here, looking as one of the requirements, you have to look at the xylem and the phloem, and the root as well as the shoots. So these are two areas that you have to know how to spot them, because these pictures here can't show up in a B exam. Now, we have here on the root, as you can see, usually in the middle, as you can see that it's been circled, xylem is closer to the middle and the phloem is more on the outside, the little blue ones. So once again, you can see that the blue ones are the phloem, and on the inside is the xylem. And again, here on the dicotyledon, which is another type of plant, you can see the xylem is usually on the middle and the phloem is always on the sides. So this is a way that you can spot. When we talk about the stem, again, you can see here the little circle, and the phloem is always on the outside and the xylem is more in the inside. Same thing here, xylem's on the inside and the phloem. So basically, if I had to kind of draw here, these bottom part would be the xylem going on, as you can see, and then let's say put another color here would have been the phloem on the top. So it's always xylem more to the inside and phloem more to the outside. If you can remember that, then your chances of getting it wrong will be very much less. So now let's talk about translocation. When I say the word translocation, I am talking about transferring nutrients in the plants, and they usually can go up or down. Now, translocation includes two things. They include a xylem and they include the phloem, but the whole process really it is happening in the phloem. Although the xylem will kind of help, translocation is for the phloem. Now, in the phloem, of course, we have two places that it goes to. One of them is basically the leaf, And then the other one is basically it can be the roots or it can be a fruit. Now, what does that mean? So here, the leaf will be the source of sucrose. That means the leaf is the one who does the photosynthesis. It is the one that produces the sugar. In this case, we're talking about sucrose. Now, when I say sink, that means it's a place where it has the storage, and the storage will be fruit or the root. So you do one of the first things you need to know is source and sink. These are two areas in which the sugar that is produced in the plant will play around. Knowing that the source is the place where you actually produce the sugar, and sink is the place where you storage. So that's why right here you can see that there is a leaf in this area, and then in the bottom you have mostly can be a root or fruit. Now, what is the phloem doing in this process? So the phloem right here, its job is to basically transfer that sugar from one to the other. You're going from the source to the sink. That's what you're trying to do. And what does the xylem have to do this? Well, xylem will help with the water transfer these nutrients going from the source to the sink. We're going to talk more in details how these guys go, but here is just to get a little bit of the exposure of this whole process. Now, also another thing that I want to give you already starting awareness is basically when we talk about water, water will always move through osmosis. It's very important to remember this. Water will always move through osmosis. When the sugar in this case, which is sucrose, will always move through active transport. So you can see here, there's a red right here. Let me put in the highlights so you can see there's a red here, and that's the sugar going from the leaf into the phloem. And you can also see a red here, but it's very much vague. It's also the sugar going from the phloem into the fruit. But when it comes to water, water is with the blue, which water is always moving through osmosis here, or even coming back here will be osmosis. So water is always moving through osmosis, and the sugar is always moving into active transport. And right here, I will highlight in blue right here, as well as here. There's companion cells, right? Companion cells is on the side, while you have the sieve tubes right here in the middle, right? This middle part here, all of this is the sieve tube, and here on the sides, this is the companion cells. The companion cells, if you remember, it has mitochondria, so it's producing that ATP. Why do you need the ATP? Obviously for what? For this, active transport. That's why it's right here, these guys. So this is just getting an introduction translocation. I'm also going to draw this more in detail and explain step by step. So you're going to eventually see this picture, but this is just to start an introduction. So first, let's talk about the source to the sink. We know that the sources are going to be placed where it's going to be produced, right? And then they will produce and send it to the sink. So when we talk about sink, we are talking about places where you can store them. So it's more the storage, and source is more of a production here. So in this case, we also know that the source will be usually the leaf, because that's where the photosynthesis is happening, and that's where the production is happening. And storage in this case can be the root, or it can be the fruit. These are the places where you can store the sugar that is produced, which is literally basically here. Source place where the food enters, and sink is where the food is removed. So both source and the sink are attached right next to the phloem. Now, when we talk about nutrients that can be transported through the phloem, we talk about sugar, amino acids, plant hormones, and small RNA molecules. All of these are nutrients or hormones that are passing through the translocation of the phloem. Okay? So right here, I'm going to explain the process of translocation, but I'm going to use the whole process by first talking about the phloem. So we have here the phloem, and if you have the phloem, we have the sieve tube, right? Because that's one part of the phloem. And then the second part of the phloem is what we call the companion cells. So what we have here is that we can have nutrients going up or down, this way or that way in the phloem, as long as they're going to the direction they need to be. Now, these companion cells, they usually have a nucleus. But besides the nucleus, they also have, let me put it here in another color, orange, they also have mitochondria. Now, what's the point of the mitochondria as you hear? These mitochondria is basically production of ATP. And why do you need ATP? Because literally in these companion cells, they have these black little dots here that I'm going to show here. What are these? These are membrane proteins that are basically active transport. And if you remember, active transport can only transport things using ATP as energy. Now, right next to the companion cells on both sides, this one on the top here, I will create this as a leaf. Why? The leaf, because this is where the photosynthesis is happening. And down here, I'm going to make an apple, so I'm going to put it in orange, and because here is where you're going to have the sink, so we can say this is the sink. So this can be a fruit or a root, right? And usually this is where the storages happen, where that means you hold on to all the sugar that's being produced from photosynthesis. So the leaf is producing that glucose and then eventually sucrose. So I'm going to put here all these dotted lines here as green to represent glucose or sucrose. Now, these ones that are producing the leaf, their job is to eventually to get here to the phloem. The question is, how do they move through the companion cell? Now, the motion that they're going through this, I'm going to put in purple. That means to pass through all of this, and I put a line in purple here, and I'm going to use the line purple here to say that it's through active transport. Right? So that means the sugar is moving from the leaf and the sugar is getting to the phloem. Okay? That's the pathway. Once the sugar gets down here, it's the same thing. The sugar will move the same way through active transport, passing through the companion cell, right, which is right between them, to get to the fruit or the root and then be stored there. So in both cases, active transport. But the question is, how does the sugar go down there? That's the question. How? That's where we have the xylem there right next to the phloem with a job. So the xylem here, if you remember, it's just a movement of upwards of water, right? Xylem just pushes the water up. But if you remember from topic 2, then if you have a lot of high concentration here of water, and here on the phloem, you have a high concentration of sugar, right? So what does this mean? When you start having so much sugar here, you start increasing high concentration of what? Of solute. And if you remember, water is always attracted to a place of high amount of solutes. So what does the water do? The water craves to go to this side so it can be surrounded by all of this sugar. So it does this by going through osmosis. So the water moves from the xylem into the phloem. And now what we have here, we have this known as a bulk or a mass flow that is going to happen. Why? Because imagine if you have a sand of a bag, and if you put water inside that sand of a bag, what's going to happen? The bag is going to get more heavy, right? You've probably seen places where, there's hurricanes and whatever, and I don't want to say tornado, but places where there's like flood going, so they make those sandbags, and those sandbags has a lot of sand inside, and when it's water on it, it gets very heavy. So this is it. It gets so heavy that it pulls not only the water going down, but also the sugar. So the sugar goes down, and eventually the sugar gets to the bottom. Now the bottom, once it gets here, what's happening? They are going to want to go in direction towards the fruit or the root to get stored. And when this happens, what happens to the solute here? You're going to see it decreases. So what you're going to be left with? You're going to be left here with just water. Now, obviously, some water goes inside as osmosis to the fruit. That's why some fruits have water. But then you're going to see that here there's a high concentration of water. And here, if this water goes up, for that 1 second of an instance, there will be no water exactly at this location here, right? There will be no water here. Why? Because the water has moved up. When for that 1 second, here, 1 second, the water here is going to be low, and the water here is going to be high. So what's going to happen? The water is going to go from a high concentration to a low as osmosis. So the water will come back to the xylem, and then again, it will be continue to pull up. And why it's pulling up? Because of transpiration, right? Which, if you remember transpiration, it has to do with the hydrostatic pressure. What is mean by hydrostatic pressure? Is that this pressure is pulling eventually the water next and the water. So as one pulls, the other one pulls after it. And that water keeps pulling them up. So this process keeps repeating all the time. So once again, you have the leaf here, right? The leaf is producing through photosynthesis sugar. It moves through active transport all the way to the phloem with the help of the companion cell, because the companion cell is producing ATP with the mitochondria. So all that sugar gets up here in the phloem. So now you can see a bunch of solute in the phloem that attracts the water in the xylem. So the xylem water goes from the the xylem to the phloem through osmosis, gets here. Now you get a heavy weight of solutes, sugar, right, with water. So that causes this mass flow or this book flow of sugar and water to go down. Once they get here, all of the sugar goes and gets stored in the fruit or the sink, and they move through active transport. Again, the companion cell right in the middle here, happening with the mitochondria, giving ATP. And then the water here will be in high concentration to low concentration of water for that one instance. That will cause the high water here move through osmosis back to the xylem, and then this process keep repeating, and you can see that there's nutrients moving up and down and being stored or produced. So that's what translocation is all about. So one more thing that we need to understand here, a little bit more detail, since now we understood, is here we have the phloem, as we have learned, and the companion cell we understood that is right next to it. And you can see here you have the mitochondria, right? The mitochondria is right here. Now, if you remember, the mitochondria, the whole job of the mitochondria was to produce ATP for the whole process of active transport. Now, we know that the nutrients, such as when it comes from the leaf here, right, will go through the companion cell and get to the phloem. Or it's going to go, the nutrients of like the phloem is going to go through the companion cell to get to whatever, the sink. Now, this passage that they're doing here, these are active transport. Now, they are basically what we see on this picture right here. You can see right here that this is an active transport. And you see that because you have ATP using for the channel to open it up. So with that happening, we have hydrogen moving, and then hydrogen coupled with the sugar is moving towards the phloem. So basically here in a nice drawing, let's put it here, and let's put here the companion cell. This is a little bit more detail, just going back. What am I doing now? I'm talking about what's happening right at this area, okay? So I'm talking about more in detail what's happening with the companion cells in the phloem. So we have here the phloem, right? And the companion cell has its nucleus, it has its mitochondria, right? And it has its protein channels. This protein channel is basically this stuff here. So what happened is you got hydrogens, or we're going to say protons, found in the phloem by themselves. What do? They move into the companion cells. And when they're in the companion cells, and you have, for example, the sugar coming from the leaf into the companion cells right here, both hydrogen and the sugar moves back into the phloem. Another hydrogen sugar moves back to the phloem through these active transports. So it's coupled, the hydrogen with the sugar is coupled together through the channel to move to the phloem. But for this to happen, you need the hydrogen to come into the apoplast, or in this case can be into the companion cell, and then eventually go in. So what is this process all trying to tell you? Is that it's not just an active transport pumps the glucose by itself to the other side. No. What this only means is very simple up, is that every time you want to get a sugar into the phloem, right, the sugar has to couple with hydrogen to move to the phloem. And this can only happen if some hydrogen move to the companion cells to match themselves with another sugar, and then they move. That is the process of active transport, and that's the process of getting the sugar from the leaf eventually to the phloem. So here is just showing the picture again in the beginning, and this time, now that you kind of got a little bit more understanding, now you can understand the drawing. So in the drawing, remember we had the xylem here, and you see how here we have low hydrostatic pressure and then high hydrostatic pressure here. So that means there's a high amount of water. Now here on the other side, we have the leaf. And if you remember, the leaf is where it has the source. This is where photosynthesis is happening. So then the photosynthesis that is happening causes you to produce a bunch of sugar. Now the sugar moves into the companion cell. And if you see the red here, what is the red? Active transport. So the sugar moves into the companion cell and eventually will move into the phloem. Here, when you have a lot of sugar, what's going to happen to the water? Through osmosis, the sugar will mix with the water, and the buck flow will drag it down. Once you get here, you see the red, again, active transport. The sugar gets into the fruit, in this case, as well as some water, But because there's still a high amount of water here, it will move to a place which has lower amount of water, and it goes back by osmosis, and then this pathway just keeps going. So what are the important things that you have to remember about these? It is simple that you need to know the sources are basically the leaf, and the sinks are such as the root and the fruit. You also have to mention how the active here, active transport, is helped by the companion cells, which have mitochondria to produce ATP. We also have to talk about how the xylem right here is going to help with its water through hydrostatic pressure to move this way for osmosis, and back here by osmosis, and eventually cause a mass bulk flow and cause the flow of sugar going down. So if you understand that there is an osmosis going on, and at the same time there's active transport going on, and you know which way their pathways are going, you actually got the translocation really good. So if you want to start the process of writing, always start with talking about the leaf, the production of sugar. Second, then talk about the companion cell. And when you're going to talk about companion cells, the most important thing is mention about mitochondria. Mitochondria and the active transport channel, okay? Then after this, talk about the phloem, as well as the sugar that causes the high amount of water to move towards the sugar, right? And this is where you can also talk about the osmosis, and then talk about the bulk flow that is going down. And then 5, again, the sugar moving, the sugar moving to the fruit, again through active transport. And then 6, the water going back to the xylem through osmosis again. So these are the main ideas. This is very outlined just for you to remember the steps. So make sure you are mentioning photosynthesis, companion cells with the mitochondria and active transport. Make sure you're talking about how the sugar in the phloem causes the water to go in, so it becomes osmosis. We have the bulk flow going down. Talk about sugar moving back to the fruit to active transport, and how the water going back to the xylem to osmosis. If you mention this pathway, there is no chance of you losing points. Not counting, of course, talk about the source and the sink. So here's another picture that you might get, and it's from BioNinja, which is even more clear, but by now you should really start to spot, right? What are you spotting here? You can see the source, right? It's the plant leaf, and you can see, for example, here saying leaf cell. And what are you doing here? You produce the sugar, which are the red dots, right? These red dots will pass here. And now what are these red colors here? Active transport. And you can see there's a companion cell between them, right? Between the source and the sink with the phloem. Sugar moves here. This causes the water to move this through osmosis, and then it goes down. And you can see through active transport, the sugar is going towards the sink. And the water, of course, goes back through osmosis. So if I had to write here, I would put osmosis here and osmosis here. For who? For the water, right? The water is always moving through osmosis. And then here we can talk about photosynthesis. And here we talk about storage, right? And these examples, we can talk about the root and the fruit, not just the root like it's written here. And this, we have the leaf, of course. Then the companion cells, we can talk about having the mitochondria, which is not shown here, but what does the mitochondria give? ATP. Why we give ATP here for? Because again, these guys here, what are they? They are active transport. So you can see that by now, you've seen this as a third time, so it's becoming more sync in your head. And hopefully you're taking notes of what I'm saying, so it becomes even stronger in your head and you're able to explain this process. So again, now it's not pictures. I have it written, so it makes more sense. When I say mass flow theory, that's basically translocation. The load, when I say loading, that means I'm putting the substance somewhere. So sucrose is actively transported from the source, water potential decreases inside, water enters from the phloem by osmosis, increased hydrostatic pressure inside the phloem near the source. Now, when I say hydrostatic pressure, it is the pressure that the water is putting there. And that pressure is what pushes the sugar to go down. Then the unloading that means getting the sugar out of the phloem into the source. So sucrose is actually transported into the sink. Sorry, I probably said source, but it's a sink here. Water potential is increased inside. Water leaves the phloem by osmosis and decrease the hydrostatic pressure inside the phloem near the sink. So that means the chances of the water moving back is going to be higher, so it's pushing it towards the xylem. So then mass flow, the loading, unloading cause gradient in the osmosis pressure inside the phloem, cause mass flow of substance down from the source to the sink. The whole point is we are manipulating the water movement by putting that sugar in the phloem. So there is an attraction of the sugar in the phloem that causes the water to be attracted to it and move by osmosis into the phloem, and that causes a hydrostatic pressure. Then when we put all that sugar inside the fruits or roots, that water will go in direction towards the xylem back to its place because the xylem again will have a low pressure, and that means the high pressure left from this phloem with the water will go back. So that's what translocation is. Now we're going to talk about an experiment done with translocation. And once again, when I say translocation, it is the movement of sugar through the phloem, right? Going from the sink from the source to the sink. Now, when I say rate here, that means how fast the translocation is happening. So right here, one example that I'd be asked is to know about the aphid, the stylet. You have to know about this mosquito. And you have to know that this mosquito, what it has, it's basically a mouth that goes inside, which we call the stylet. And this stylet, more zooming in, this is what it does. It goes all the way, it's stuck his basically little stick all the way to the phloem because he goes and suck out the sugar out of the phloem. That's what he does. So this aphid stylet, what is he trying to do? He's basically trying to poke into the plant and try to suck out the sugar out of it. So here is the experiment that it shows. And you're just basically poking the stylet in there. And from the stylet, when you poke it and you open the hole, you're going to see the liquid that comes from the phloem is popping up. And that liquid, you can take the sugar. So what usually they do is imagine that you have a stem right here, right? We put horizontal instead of upwards. And with the stylets, we kind of hit in small spots. Like we decide a certain distance, like every 5 meters, we get this the stylet of this aphid, and we put it up here. And we poke it with his mouth in here, and we want to try to get the phloem liquid. Now, what we do is we put a radioactive carbon in there. So when we do a radioactive carbon in the plant, what happens? The plant starts producing sugar that has glucose, for example, 6,6H12O6, or sucrose. What are these guys going to have? They're going to have a radioactive carbon, and that means it's think of a highlighter. It's like if I actually highlighted a carbon. So that means these sugars will be highlighted. So what happened is the sugars are going to pass through here, right, through the phloem. What we're going to do is eventually with this, actually, let me take this out for a second here, is that as the phloem liquid is moving through, you're going to actually pop out these sugars out, right? Just like you see here. And then you never know if you have here radioactive that it pops out, and then later on it shows up again here. You can see the distance that it traveled, and you will know the time that it went through this. And you can find the fast, because the rate usually is basically the distance over the time. Or when you see him pop here, and then you figure out how much time it takes him to pop again here, you can see the distance, right, which is the 5 meters, and the time, I don't know how much, maybe it took 2 seconds or 2 meters. You put this, okay, 2 seconds here, and this is how you find the rate. So this is just to see how fast the sugar is passing through the phloem. So here's an example that they did. As I told you, they take the organism that I just mentioned, the aphid stylet, and he placed them in different places in colony 1, 2, and 3, and the starter colony, okay? Remember that they use the radioisotope, a carbon dioxide, here as a sugar that is like kind of highlighted, right? So the distance from the starter, okay, which is 1, is about 20 centimeters. The second one is 40, and the third one is 60. So this is just the distance from the start. So it's basically saying from here to here is 20, from here to here is actually, wait, from here to here is 40, and then from here to here is 60. Wait, it's a little bit crowded. Let me draw this again. So when we say distance is from the start colony to here is 20, and then from here to here is 40, and then from here to here is 60. That's what the distance. Now, time for the radioactivity to travel from a starter colony hours. Well, to go from here, for him to travel from here, let's say the sugar, and to get to the colony up here was 1.25 hours. And now to get from the starter to all the way to the colony, there was 2.40 hours. And then to get all the way to here, it took him 3 hours and 80. So this is just finding time. Now, since you have the time and the rate, as you can see here, distance over hour, right? Because this is -1. So they did the calculation. The rate here is 16, 16.7, 15.8. So you can see the rate is basically on a 16 averagely. This is how you can find the rate of translocation. So business process here is just to wrap up that you can use the rate of calculation. And so once again, translocation, this is something, it is the movement. It's the movement of sugar going from the source to the sink, right? And in this process, of course, you're going to have the phloem included. We are going to have the xylem. And from the phloem, we're going to use the sieve tube as well as the companion cell. And the xylem, of course, we're going to use the hydrostatic pressure of the water. Now, besides that, we're going to have the motion of osmosis and the active transport. So this is a very generalized. And then if you want to do the rate of translocation, of course, you want to find the rate by doing the distance over the time. And you do this by looking at stabbing at different points of the stem cell, or the stem of the plant. And this is doing with an aphid, right? Use the stylet of the aphid. And from there, you can find the distance as well as the hour for this, and you can find the rate of translocation. So the rate of phloem will principally be determined by the concentration of the solid sugars in the phloem. Of course, it all depends on how much sugar is in the phloem, okay? Because if you don't have enough sugar in there, you cannot identify it. Now, the concentration of the solid sugars in phloem sap will be affected by what? The rate of photosynthesis. So yeah, if you have more photosynthesis, that means you're going to produce more sugar. But what makes the photosynthesis go more faster? More light intensity, more CO2, depending on the temperature, it can increase in the beginning. The rate of cell respiration. Don't forget that even plant can do cell respiration. That means the plant can actually use glucose for its energy. Now, the rate of transpiration. Well, sometimes you have a lot of the water leaving the plant and not much water is going to go through the phloem. So that means it can slow down the phloem. And the diameter of the sieve tubes, that means how big is the phloem? Sometimes it can be very small, it can be large, and that can affect the hydrostatic pressure. So these are all ways in which you can manipulate the rate of reaction. So don't just think is the question, oh, sugar just passing through. No, we have to see the photosynthesis effects, the cell respiration, the rate of transpiration, and diameter of the sieve tubes. These 4 things can affect the rate of phloem. So you must be knowing this. This might happen as a multiple choice question or even an essay where you have to talk about these guys. And that's pretty much for topic 9.2 on phloem translocation. Here are the study guide questions. Practically, my video answered most of these questions, if not all. If you can answer these questions, then you have mastered this entire 9.2 topic. And that's pretty much it. Thank you.